Denaturation refers to the process where a protein loses its natural three-dimensional structure due to external stress or conditions, resulting in the loss of its biological function. It affects the secondary, tertiary, and sometimes quaternary structure of the protein, while the primary structure, amino acid sequence, generally remains intact. Causes of denaturation. 1. Heat. High temperatures increase the kinetic energy of the protein's molecules, breaking the weak interactions, like hydrogen bonds, that maintain the protein's folded structure. An example is cooking an egg. The heat causes the egg white proteins, like albumin, to denature and coagulate. 2. pH changes. Extreme pH levels can disrupt ionic bonds and hydrogen bonds by altering the ionization state of amino acid side chains. This can occur in cases like acid reflux, where the acidic environment can denature digestive enzymes. 3. Chemicals. Organic solvents, like ethanol, can disrupt hydrophobic interactions. Urea and guanidine hydrochloride can break hydrogen bonds and destabilize the protein's folded state. Detergents can disrupt hydrophobic interactions within the protein. 4. Radiation. Ultraviolet or ionizing radiation can damage proteins by disrupting bonds and inducing abnormal folding. 5. Mechanical agitation. Vigorous shaking or stirring can lead to protein unfolding by introducing physical stress. Consequences of denaturation. Loss of function. Since a protein's function is determined by its structure, denaturation typically leads to the loss of biological activity. For example, enzymes lose their catalytic ability, and structural proteins lose their mechanical strength. Reversibility. In some cases, denaturation is reversible if the denaturing agent is removed. This is known as renaturation. However, in many cases, especially with extreme conditions, the process is irreversible, leading to permanent loss of function. Examples of denaturation. 1. Cooking proteins. When you cook meat or eggs, the proteins denature and coagulate, changing their texture and appearance. 2. Fever. High body temperatures during a fever can potentially denature enzymes, impairing normal cellular function if the fever is too high. 3. Disinfection with alcohol. Alcohol denatures the proteins in bacterial cell walls, leading to cell death, which is why alcohol is used as a disinfectant. 4. Milk curdling. When milk becomes acidic due to bacterial activity, casein proteins denature and precipitate, forming curds. Importance of denaturation. Understanding protein denaturation is crucial in various fields. Biology and medicine. Conditions like high fevers or acidosis can lead to protein denaturation in the body, affecting enzymes and other proteins vital to physiological functions. Food science. Cooking, fermentation, and food preservation often involve controlled denaturation of proteins. Pharmaceuticals. Protein-based drugs, such as insulin, can lose their effectiveness if exposed to conditions that cause denaturation. Denaturation plays a critical role in processes ranging from food preparation to biological function, highlighting the importance of protein structure in maintaining proper activity. The reversibility of denaturation depends on the extent of the structural changes and the nature of the denaturing agent. In some cases, proteins can regain their native structure and function through a process known as renaturation, but this is not always possible. Factors influencing reversibility. 1. Extent of structural damage. Mild denaturation. If the denaturation is mild, e.g. due to moderate heat or slight pH changes, the protein may refold into its original structure once the denaturing agent is removed. Severe denaturation. If the denaturation is severe, such as exposure to extreme heat or chemical conditions, the damage to the protein's structure may be too extensive for it to refold correctly. In these cases, the process is irreversible. 2. Type of denaturing agent. Heat. Heat can often cause irreversible denaturation because it can lead to the formation of aggregates, clumps of proteins that are difficult to unfold again. For example, once egg whites are cooked, the protein aggregation is irreversible. pH changes. If the pH returns to normal after a mild deviation, proteins may renature. For example, some enzymes can regain activity after being returned to their optimal pH range. Chemicals. Some denaturing agents, like urea or guanidine hydrochloride, can often be removed, and proteins may refold correctly in their absence, restoring function. However, strong detergents and organic solvents often cause irreversible damage. Number 3, protein structure and folding. Small, simple proteins. Proteins that are small and have simpler folding patterns are more likely to renature after mild denaturation. These proteins can spontaneously refold into their original structure once the denaturing conditions are removed. Complex, multi-subunit proteins. Large proteins with complex quaternary structures are less likely to renature successfully. The more complex the folding and the number of subunits, the more challenging it is for the protein to refold properly. 4. Chaperone proteins. In cells, special proteins, known as molecular chaperones, assist other proteins in folding or refolding after denaturation. These chaperones help prevent misfolding or aggregation during the renaturation process, improving the likelihood of correct folding. Examples of reversible denaturation. 1. Ribonuclease A, RNase A. This enzyme, a classic example, can refold and regain its activity after denaturation if the denaturing agent, such as urea, is removed under appropriate conditions. Its renaturation shows that not all denatured proteins are irreversibly damaged. 2. pH-sensitive proteins. Some enzymes that are denatured at acidic or basic pH can refold once returned to their optimal pH environment, such as certain digestive enzymes that function in the stomach or small intestine. 3. Reversible gelatin formation. Gelatin, denatured collagen, can be reformed when cooled and melts again upon heating, which is an example of reversible denaturation, though this is not representative of functional proteins in a biological system. Examples of irreversible denaturation. 1. Egg cooking. When egg proteins like ovalbumin are heated, they denature and form a solid structure. This process is irreversible because the proteins form aggregates that cannot refold. 2. High fevers. In humans, prolonged exposure to high temperatures can cause irreversible denaturation of vital enzymes and proteins, leading to cellular damage and possibly death. 3. Alcohol denaturation. When alcohol is used to disinfect surfaces, it causes irreversible denaturation of bacterial proteins, effectively killing the bacteria. Importance of reversibility. In biotechnology and medicine, understanding reversible and irreversible denaturation is crucial for developing protein-based drugs where stability and activity must be maintained. In enzyme function, many enzymes undergo slight denaturation during normal activity, but their ability to renature is vital for continued function in the cell. In summary, the reversibility of denaturation depends on the severity of the denaturing conditions, the type of protein, and whether appropriate conditions for renaturation are restored. While some proteins can refold and regain function, many undergo permanent structural damage, leading to irreversibility.